Appreciate the invitation to be here and to talk a little bit about this topic, which is sex-sorted semen. Of course, we've already talked about that a little bit. And so Dr. Corder has gotten to share some of the really fun applications of it and those really business things played out in terms of how it all makes money. My focus today is really not going to be that piece as much. I'll talk a little bit about some of those concepts here as we get going. But I want to talk about if you were going to use sex-sorted semen, how could you actually potentially do that and actually get good pregnancy rates? That's something I've been really interested in for years now. And when I was a graduate student, to think about the kind of pregnancy rates that we're getting now, not just with conventional semen, but specifically with sex-sorted semen, was just unheard of. I mean, I remember some of the first trials that I did with sex-sorted semen and getting pregnancy rates in the high 30s and going, well, okay, yeah, that kind of makes sense. And I would be really disappointed by those outcomes today, and I think many of you in industry would too. And so we've made a lot of progress. Let's start here, though, and just talk a little bit about the business stuff here as we get going. So the University of Missouri Extension every year puts out a cow-calf planning budget, and they're always wrong because all projections are always wrong. But I always find it fun to look through that publication every year and see some of the assumptions that are made. And so there's assumptions made for both fall and spring calving sort of production models. And they're way wrong about the calf prices. What they had in there for price per hundredweight for 5-weight steer calf started with a 2. So it tells you how wrong they were compared to maybe what many of you are selling calves for today. But if I show you just the operating costs that are assumed in that sort of budget, that planning budget, I think we all know what the number one direct cost of production or operating cost of production is associated with every calf that we produce, and it's feed, right? It's stuff mostly that the cow eats, and that point's already been made a little bit today, but that's our number one piece of the pie. You know, if you cut up a calf into a pie chart and you tried to represent the costs associated with producing that calf, feed-related costs, if you include pasture access, are over half of that pie for most people's budgets. But the second biggest piece of that pie, and a huge piece, is this cost here of replacing cows that become open during that next breeding season. That is just a huge, huge piece of that pie, and you see that represented in this budget in the $400 range per calf produced. And I love the way that Trey Patterson, the manager of the Padlock Ranch, talks about this particular challenge. He says, of course it's your second biggest cost of production. Imagine if you had a factory, we've used this analogy already today, imagine if you had a factory and every year 20% of your factory burned to the ground and you had to rebuild it, right? That would be a huge cost of production, and that's basically what happens in our cow-calf herds when we have high replacement rates that cause high replacement costs. So before we ever talk about sex-sorted semen, let's put this little lens on it. If you think about a 60-day breeding season that leads to a 60-day calving season and then follow that with another 60-day breeding season, I just always try to make this point, just as context. If gestation in cattle is 283 days and the year is 365 days long, this animal that calves on the first day of the calving season, because she conceived on the first day of the breeding season, she's got about 82 days before that next breeding season starts. And that 82 days is a period of time in which she's going to go through what we call postpartum estrus, which is basically that at the level of her brain, there's a big brake pedal placed on reproductive function, and that brake pedal's got to come off. Well, you consider a cow that conceives on the last day of a 60-day breeding season and calves on the last day of a 60-day calving season. Well, she doesn't have 82 days anymore, right? She's got 22 days until that next breeding season starts, but she's still got to go through all of that same physiological process of postpartum estrus, and she's not going to get it done in 22 days. And so there's a couple implications, just to think with our business lens here for a second. There's a couple implications of this. That cow at the bottom doesn't just magically become the cow at the top next year. It just doesn't happen. Now, I'll say that having managed a lot of cows over the years, it does happen every once in a while. Okay, I'll give you that. Okay, but if you have 100 cows, okay, that are set up to do that, 100 of them aren't going to switch like that. Maybe 1 or 2 do, right? So these later conceiving, later calving cows don't just magically become earlier conceiving cows next year. If anything, the trend is they move later, right? Because they're behind. The other implication here is that they wean the sorriest, lightest calf in the group next year, right? That cow that conceives late. So that's important to note. But which cow here drives up our second biggest cost associated with every calf we produce? It's obviously the later conceiving, later calving cow. So before we ever talk about sex-sorted semen, we got to think about how high this bar is for a replacement heifer. And those of you that were out at Cattle Visions yesterday, we talked a little bit about this already. But this is some actual commercial data. So this is some commercial herd data out of herds in South Dakota. This was in a nice publication by Bob Cushman and others in 2013. And the black line there, the solid black line, is the retention rate of those heifers that conceive in their first 21 days of their first breeding season and calve in their first 21 days of their first calving season in the herd over successive years. The dashed line is those heifers that conceive later than that in their first breeding season and calve later than that in their first calving season. A couple things to note here. It's a little disappointing the retention rate of these animals as 2-year-olds in either line. This is just real-world commercial data. I think it's around 75% or something for those that conceive in the first 21 days. That is rough. It is rough to have 25% of first calf heifers fail to breed back, right? So we want these lines higher in either case. But look at the greater rate of fallout that you have in heifers that conceive later and calve later in their first calving season. And then also look at year 3, because I think we kind of lose track of who those heifers were that calved a little bit late, right? At some point we wean that calf off and they're just cows again, right? We maybe don't put cause and effect together, but that higher fallout rate that we have in those 3 coming 4-year-old cows is also really affected by when they just conceived to calve for their very first time in their first season. And so if you go out a few years, you've lost a tremendous number of later conceiving heifers as cows. They just don't stay in the herd all that well. So I always like to talk about vicious cycles versus virtuous cycles. I think we always when we think about systems, we know that phrase, vicious cycle. It's something that gets worse and worse over time. But we can also start things called virtuous cycles that get better and better over time. It's the same kind of process of a reinforcing feedback loop, but we're pulling some different levers. And so one lever that we can certainly pull is getting heifer calves physically on the ground and born earlier in their calving season of birth. And here's a little bit of data on that. This is some data out of Nebraska Production Systems. Rick Funston and others put this together. But this is the retention rate of heifers in the herd as a result of when they were born. And the blue line is, of course, those that were born earlier in their calving season of birth. The yellow line, those that were born later. I think extension specialists over the years latched on to these kind of data and gave some advice that is okay, but maybe I'm going to take it in a different direction. You've probably heard people say something like, only develop heifer calves that were born in the first half of the calving season or in the first 30 days. And I'm not saying that's bad advice, but it's imperfect advice, okay? What if our advice became only make heifer calves as potential replacements that are born super, super early so that they're old enough, right? That's an interesting spin on that piece of advice. But we can do that now with sex-sorted semen. What if you think about the production models that Dr. Corder already talked about, maybe the piece to emphasize there that we didn't emphasize enough already is those heifer calves are now going to be older when they start their first breeding season, and that has some major impacts. Here's why that last slide looks the way that it does. If you dig into that data, the heifer calves that were born in the first 21 days were more likely to be cycling at the start of their first breeding season and themselves go on to conceive in the first 21 days of their first breeding season. Therefore, they become that earlier calving animal that we just talked about in the first couple slides. So major opportunities. I should also note, this is kind of a lower input production context. This is a little bit of a what we would call a 55% target weight kind of heifer development model. These heifers are not super pushed hard, and so this is maybe the most exaggerated that those differences could be, but I still think that's something that we run into, is the challenge of the age of heifers particularly if they're not super pushed hard developmentally. So I have some USDA funding right now in the space of sex-sorted semen, and we've made a lot of progress on this already, but I want to just set the stage and talk about sex-sorted sperm cells here and why they may be a little bit different. So when I teach reproductive physiology to our undergrads, I'll talk about how the head of the sperm cell basically is not just this boring head of the sperm cell. It's got these projections sticking out of it, these proteins and other molecules, signaling molecules and things like that. And also the membrane dynamics because of the presence of cholesterol versus maybe lower content of cholesterol. It's got some differences in its fluidity that allow some different things to be sticking out. Okay, so to oversimplify it a little bit, when those sperm cells are ejaculated out, they have a lot of surface molecules that eventually are going to get cleaved off so that it can interact differently with the reproductive tract and subsequently interact differently with the oocyte. What happens with sex sorting is sort of like what normally happens with capacitation, this process of sperm cells acquiring the capacity to actually fertilize an oocyte. What normally happens in the tract with sex-sorted sperm cells, often a larger proportion of them have more and more of those important molecules cleaved off of them. It's not necessarily bad because they would go through a process like that anyway, but it changes our useful lifespan. So why in the world do I have an image of waves here on the side? So this concept of waves is actually pretty helpful. One way that you could think about this is in a normal ejaculate, some of those sperm cells have already had those molecules cleaved off and have already had some of these changes that give them the capacity to fertilize. And that's actually important because if that female is going to ovulate in just a few minutes, believe it or not, there are mechanisms that get sperm cells to the site of fertilization in just minutes. And that is part of what we know occurs in biology. But then we have additional waves of sperm cells reaching the oviduct later, more like in the hours range. And those sperm cells, kind of in this other wave, they still have all of those molecules on them, and they have to go through this process of capacitation that gives them the capacity to fertilize. And so the more variation that you have in the status of those sperm cells, essentially the more waves that you have, it's not a perfect analogy, but the more waves that you have that sort of cover a broader period of time. And what happens with sex sorting is basically you have a lot fewer waves, you have a lot less variation, more of those sperm cells are driven to be more like what we would call precapacitated. And so that's a challenge, because if you inseminate shortly before ovulation, that's maybe not going to have that big of an impact. But what if you inseminate super in advance of ovulation? Now we don't have waves of functional sperm cells capable of fertilizing at the appropriate time. And so this is a little bit weird, right? Because we're not just talking about fertility high and low. We're talking about fertility high and low and over time. And that's the challenge with sex-sorted semen. So we know that sex-sorted sperm cells face a quality issue, and that remains a challenge. And so we're probably not going to overcome that challenge with anything that my lab does, because we tend to focus on the cow and how we manage the cow. But we do know that there are some challenges even in these in vitro systems with blast rates and cleavage rates and so on and so forth. There is a little bit of older literature that would say fresh sex semen is advantageous, but for the most part, the companies don't want to mess with that, and for the most part, producers probably also don't want to mess with the logistics of getting fresh semen to them as opposed to frozen semen that comes in a tank. So just some logistical challenges there. Another thing that we really propose to address in this grant, and that we've honestly really struggled with, is addressing the fact that there are bull-to-bull differences in fertility with sex-sorted semen that we, as of yet, don't do the best job of characterizing or really even understanding. And so just because a bull has high fertility with conventional semen, believe it or not, it doesn't even mean that he will successfully sort and freeze semen that is viable. And so there's no guarantees there. And so some bulls and some ejaculates from some bulls just don't successfully sort and go through the freeze-thaw process successfully. And so there's also potentially some differences in bull fertility when using time-to-eye versus heat detection, although we really haven't been able to prove that yet. because it takes just big numbers to do so. So in terms of trying to get to an actual application, how do you actually use sex-sorted semen effectively? Here are a few things that we actually do know today that could get you some better success rates with sex-sorted semen despite some of the mysteries that remain. First is that when estrus expression occurs matters, and here's why that is. I think we all forget this a little bit, but ovulation occurs quite a long time after the onset of behavioral estrus in cattle. It's actually closer to 30 hours after the onset of estrus. And so estrus itself in cattle is a variable length of time. It's going to vary from female to female and even from expression to expression within female, but it's not uncommon for estrus to be in the 8 to 12 hour range. But again, that LH surge that initiates the ovulatory process happens around the onset of estrus, and we're not going to have ovulation until about 30 hours later. That's quite a long time, and so that influences some of the biology that we're up against with sex-sorted semen, where those sperm cells have a reduced longevity within the tract. So when estrus expression occurs matters. Also, whether estrus expression occurs matters. That matters a lot when we start to talk about timed artificial insemination systems and how to actually get good success in a somewhat easy way that doesn't involve just heat detection for a period of several days. So when estrus expression occurred mattered, and whether estrus expression occurred mattered. We're working a lot with the when piece right now, and so fortunate to work with a company called Cow Manager, which Select Sires works with quite a bit, and we've had some really nice partnerships with Select Sires and Cow Manager on this data acquisition. But trying to actually get data collected on a large number of animals to have a very specific known onset time of estrus expression. And then we have an experimental design, which I don't know, I do have it there, where we really try to, because of some of the math that we did in planning this, really try to have a broad variation in the timing at which AI is performed relative to the onset of estrus with either conventional or sex-sorted semen from the same bulls. Just a really cool design with a couple of protocols, but I don't have a lot to share with you yet. Another thing that we're doing is we're collecting some genetic samples on those animals, and actually could potentially at some point look at some of these genetic differences that may be driving estrus behavior to be greater intensity or less intensity, and so on and so forth. One thing though that we do know now that is really effective is the use of an estrus detection aid. And so if you're going to use sex-sorted semen, and really in a lot of ways, if you're doing AI at any scale, I would always recommend using these estrus detection aids to understand what's actually happening. I think they're, for the most part, inexpensive, and it can increase the accuracy of what you're doing, and it can allow you to do some timed AI strategies that are really interesting. And so here's maybe one of the reasons I say to think about doing that. This is some data from a colleague of mine, George Perry, who at the time that this was published was at South Dakota State University. Many of you may know George or may have known George when he was there. He's now at Texas A&M University at an outlying station and does a lot of really nice research. But even in this study with a 7-day co-sync plus cedar several years ago, there was a pretty strong effect, of course, of the type of semen used. He calls it gender skewed in this study. It's the same as sex-sorted semen. It's the same thing, just a different terminology used in that particular paper. But you see reduced pregnancy outcomes with that sex-sorted semen compared to conventional. But what's really interesting is if you look at what's going on with the Estrotec patches that were applied to those animals, when those animals had a non-activated Estrotec patch here over there on the far right, they achieved substantially reduced conception rates to artificial insemination when sex-sorted semen was used compared to if conventional had been used in those animals. So there's always a bit of reduction regardless, but we know that that reduction gets really big when we use it in an animal that has a non-activated estrus detection aid at the time of a fixed time AI. And that goes along with some data that I had collected as a graduate student as well, where we were developing this split time AI approach that I'll show you here in a second. But so we know that just in terms of certain recommendations, if you're going to use it in a heat detection program, let's say that you're detecting estrus over several days and you're thinking about using sex-sorted semen, what do you actually do that's different? Here's what I would do that's maybe a little bit different. Devote time to checking those animals at least 3 times a day instead of just twice a day. And the reason is our best recommendation with AI with sex-sorted semen following detected estrus is to be closer to the 18 to 24-hour time window after detection of estrus for the actual AI to occur, rather than maybe that 12-hour time window that might be a little more acceptable with conventional semen. So we often talk about this AM/PM rule, and if you are thinking through the logistics of it, if you go out and detect an animal in estrus in the morning with conventional semen, you would breed her that evening. That's still probably the right thing to do with sex-sorted semen because she may have come in heat right when you went back to the house the previous night, right? And so she could have been in heat for more like that 20-hour range. And if we were to delay her to the next morning, that would be too late. But by going out and doing an afternoon check, you get a little bit more information about those particular animals and whether they, when to actually successfully perform insemination on them. That allows you to do those animals in the following morning. And then those that are in heat that evening, you could actually push to the following evening and be at a more acceptable time for those animals. And so it just gives you a little additional precision. I think more and more, though, people are just moving to timed AI systems because they work. And timed AI really works effectively, and it doesn't require the same kinds of labor. And you can, today, use sex-sorted semen effectively in some time programs. So some examples of those here. I would consider only using sex-sorted semen if they have an activated estrus detection aid, and having some conventional semen on hand in a commercial case, or having some different sires that you have conventional semen on in a seed stock case, depending on what you're trying to accomplish. But using that only in animals with activated estrus detection aids. We know that pregnancy rates are always lower in non-estrus animals, even with conventional semen, but it's even larger with sex-sorted semen. I throw my own name up there as a reference on this because here's an extreme observation. When I was in graduate school, we had a study in which non-estrus animals were inseminated with sex-sorted semen. And I'm sure that it was a little bit specific to this bull, but we had a bull that on non-estrus animals got a 4% conception rate, despite getting conception rates in the 50 range with animals that were activated. So that tells you how big these effects can potentially be when we're talking about non-estrus animals. So it's very, very risky to use sex semen in an animal that has not expressed estrus prior to the time of AI. Here's also an example that I would give you. Use a timed AI approach where you're going to get a large proportion of the animals to actually express behavioral estrus. Not every protocol will do that. So performing AI later, like a timed AI later, you might be tempted to think that would be a good solution based on some of the things I've said today. Like if the protocol says 66 hours, let's just do 72. Don't do that, actually. There's a lot of data that says, hey, that actually, if anything, in some cases can make it worse. We've collected some data like that ourselves, and there's other publications out there. So that doesn't seem to be the right solution. Instead, just use a protocol that's really well validated with sex-sorted semen and gets a nice overall heat response. This is probably what I would do in a set of heifers. I'm a little biased because this is a protocol that Missouri invested a lot of time in, and I worked some on this split time AI piece of it. But if you look at the sex-sorted protocol sheets that get published every year with recommendations for the use of sex-sorted semen, what should you do? This protocol is on there and fairly well highlighted for use in heifers. The 14-day CEDAR PG protocol, so a 14-day CEDAR treatment as a pre-synchronization, wait 16 days, give prostaglandin and put on one of those patches, and then a very straightforward approach from there on after. At 66 hours, AI all of the animals with an activated patch. Don't need to give GnRH. There's no reason to for those animals. They're all great candidates to receive sex-sorted semen. 24 hours later, AI all of the remaining animals. If they have an activated patch, they also don't need GnRH at that time. We've validated that. And they're also great candidates to receive sex-sorted semen at that time. If they have a non-activated patch, give them GnRH. You should still install conventional semen because you'll get some of them pregnant, so it's not a waste of time. But those are the not good candidates to receive sex-sorted semen. The nice thing about that approach is if you do that in a well-developed set of heifers, there's probably only 10 or 15% of those heifers that really don't express estrus by that time. So really nice opportunity to get sex-sorted semen into 85–90% of the heifers you're working with in many cases. Here's why I say those numbers. This is the distribution of when estrus occurs after that protocol, and so the way that plays out, if you go in at 66 hours, you get about 74% of those animals expressing estrus by that time. Would service about 75% of those animals with sex-sorted semen, of course, all of those animals that were in heat at that time. About another 15% of that total group would be expressing estrus during that next 24 hours, and so they achieve some additional enhancements in their conception rate if you wait to perform that AI for them at 90 hours, so 24 hours later. And then if you were to continue heat detecting, yeah, you might catch another 5% of those heifers in heat, but for most folks, that's probably not what they want to spend their time doing because it just drags the process on. And so those are great candidates to receive sex-sorted semen, as well as those that just would have never responded in the first place. Here's some actual pregnancy rates that are published in the literature with this approach. So left side conventional semen, right side sex-sorted semen. Those that were in heat at 66 hours is the top row there. 62% if they received conventional semen, 53% was sexed. You see very strong conception rates in those animals that express estrus in the next 24 hours also. And then very similar results in those animals that were non-estrus at that time that were inseminated in that particular experiment. And so the nice thing is that even though those conception rates are very, very bad, 29% in both cases, even though those are very, very bad, you minimize the number of those animals. And in some previous work, we really showed some of those advantages that led up to that as well. I always get asked about this, but if you use sex-sorted semen, is that going to just nuke your overall into the pregnancy, into the season final pregnancy rates? In this particular study, this was about a 45-day breeding season for those heifers. And so we got 60% to conceive with conventional semen, 52% with sex semen. Then they were turned in with bulls. Excuse me, I said 60-day breeding season. I said 45. At the end of that 60-day breeding season, equivalent pregnancy rates of 89% in either case. And so yes, you take a little bit of a hit with that early conception because you're using sex-sorted semen. And so think through whether that really makes sense in your situation. There is an extension publication on this topic of split time AI, and so I'd refer you to that. And of course, it is in these protocol sheets that are published every year too. Really quick, I just want to highlight a couple things here on the cow side of things now. So this 7 and 7 sync protocol down here at the bottom left on this cow sheet is another product of my lab, and you don't see it yet on the sex-sorted sheets, but there's really strong reason to think that that is the protocol that you really need to use in cows if you're using sex-sorted semen. And here's why. Here's a slide that really shows the pregnancy rates that are achievable with conventional versus sex-sorted semen on that approach. And so if you were wondering, hey, on those slides that Dr. Corder showed, how in the world are they getting a 77% conception rate with conventional semen? That's what we published in a pretty rigorous study with 7 and 7 years ago, just a couple of years ago. And we have seen that really hold up in the field in a lot of cases. It's not always 70-something. A lot of factors have to be working right for that to be the case, but certainly possible. And then sex-sorted semen conception rates to a fixed time AI of 52% in that particular study. And so what is driving that? It's that really strong proportion of cows that express behavioral estrus before the time of fixed time AI. If you're curious about the physiology, I think I'll stay light on some of the experimental slides I share with you today on this particular approach, but here's what's basically going on. We give that prostaglandin at the front end because it allows us to create this really interesting physiology of a low enough progesterone environment that they have elevated LH pulsatility and a high enough progesterone environment that we can still keep them from ovulating. So what happens is we get those animals sent into that GnRH administration with a way higher likelihood of actually achieving a GnRH-induced ovulation. And so that really helps generate a very consistent set of animals in terms of their actual response to the overall synchronization and their overall time to AI pregnancy rates. I think I want to skip through here just a few things because I want to be respectful of your time, but I want to show you some slides with embryo transfer here, because I know some of you have a strong interest in putting in embryos. This is what happens if you use this approach to synchronize recipient cows, okay? On the left side, a 7-day co-sync plus cedar. On the right side, a 7 and 7 sync program. This was across multiple herds across the state of Kansas. 86% of those cows actually successfully expressed estrus. And that is a big, big deal because it also occurs in a very short and narrow time window. And so that expression of estrus with 7 and 7 really enables you to start thinking about fixed time embryo transfer or a reduced number of days over which embryo transfer occurs. It's not going to necessarily affect the proportion of cows that get used for embryo transfer based on palpation or ultrasound of the ovaries, because they'll still have CL either way and still get used either way in most cases. But it makes a big, big difference because you put more embryos into actual estrus recipients. So recipients that expressed estrus and expressed estrus in a narrow window of time. And so because of that, in this particular study, we don't have strong conception rate, if you want to call it that. Pregnancy rate per transfer is the more technical term. We don't have strong differences there. But if you look at the pregnancy rate per head synchronized, because you put so many additional embryos into animals that successfully expressed estrus, you overall do a much better job with recipients with that protocol. You might feel like those are a little bit low, and I agree those are a little bit low. There's a little bit of noise just from that particular year, but strong effects there of using 7 and 7 in recipients. So I think with that, I'm going to maybe leave it there. And I think we're going to move into some questions. Hopefully we got plenty of time for that. I know we'll have a great discussion.